Okay, so before we get into our examples, I do want to make sure that we know how to read a codon chart. There are two types of codon charts that you may see. The first one being this circle, and then the second one would be a codon chart that is similar to this one, where you would have first letter, second letter, and then third letter. Now, the way that you would read the circle codon chart is that you would start in the center with the bigger letters, and then you would work your way out to the outside, and that would give you your amino acid. So for example, A, U, G, you would start with A, then you'd go to U, and then you'd go to G, and that would give you methionine, or MET, which is your start codon. Right? Another one that we could do would be C, A, U. Right? You have C, then you would go to A, then you'd go to U, and then that would give you HIS, which is histidine. So I prefer using the circle codon chart, but you could see the circle one, or you could see the square one. But regardless of which one you're given, you do not have to memorize this. You just have to know how to interpret it and be able to get those amino acid abbreviations. All right. Now, the other type of codon chart is, I refer to it as the square. All right. You have your first letter on the left side, your second letter on the top, and then your third letter on the right. Right, and the way that you do this is that you just match them up. So let's just say we were going to do U, A, G. All right, so then we would go to U, and then A would be in this box. So our answer is going to be somewhere in this box, and here it is. UAG is stop. Right, and if we were going to do CGU, right, we would have C, and then you'd have G right here. So it's going to be somewhere in this box. So CGU is going to code for arginine. Right, so that's just how you would read the square one. And remember, it doesn't matter which one you're given. You don't have to memorize it. You just have to be able to interpret it and determine your amino acid from the codon that you have. All right, so let's get into some examples. All right, so I'm going to make this little small one right here. All right, so all right, whenever, like, so the whole process of protein synthesis, so I'm going to put that up here. All right, remember, is to make proteins. All right, so you're going to start off with DNA, or a DNA strand. And let's say that our DNA strand is ATG, CGA, TAT, TGA. All right, and then so we're going to need to replicate it first. All right, so replicate, remember, is you're making another copy of the DNA. All right, so A is going to bind to T, T binds to A, G binds to C, C binds to G, G binds to C, A binds to T, T binds to A binds to T, T binds to A, T binds to A, G binds to C, A binds to T. All right, then you're going to transcribe it into mRNA. All right, and you're going to use that base pair rule. So T binds to A to U, C to G, G to C, C to G, T to A to U, T to A to U, A to U, C to G, T to A. Right. Then we need to make sure that we're boxing around, starting with our AUG every 3 to make our codons. And then you would use your codon chart in order to make those that amino acid sequence. Right, so that would be translating, and that would be our amino acid sequence. And we already know that AUG is MET, right? But CGA C, G, A is going to be arginine. U, A, U, A, U is going to be tyrosine. And then UGA is going to be stop. Right? And that is the whole process of protein synthesis. Now, with the mutations that we had just talked about can either have no effect on the protein that's made at the end, or it could have an effect on the protein that's made, which would alter our traits. So let's go ahead and look at how that would work. All right, so the first one that we're going to do is going to be substitution. All right, so with substitution, and so we're going to use this original DNA strand, but we're going to make that edit to it. So remember, in substitution, that's whenever one base is going to be swapped, right? So let's say that our DNA strand is going to be ATG, then it's going to be CGA, then it's going to be TAC, TGA. So you can see where our TAT is going to be TAC now. Okay, so that's where this particular T was substituted with a C. All right, so then we're going to go through the same process of replicating it, all right, to where we're going to make that complementary strand. So then that leaves us with TAC, GCT, ATG, and then ACT. All right, so you can see where the ATA in the original strand is now an ATG. All right, so then now we're going to go to mRNA. All right, and then we're going to transcribe it. So T will bind to A to U, C to G, G to C, C to G, T to A to U, T to A, G to C, and then A to U, C to G, T to A. All right, and then we will use it to make our amino acid sequence. And we already know that AUG is MET. The CGA did not change, so that is ARG. Now, we had originally UAU, but now we have UAC. So if we look, we have UAU is going to be tyrosine, but UAC is also tyrosine. So we have tyrosine, and then we have our stop codon. All right, so this particular substitution that occurred, because the amino acid sequences are the same, that means that no change has been made to that protein, which means that the trait's not going to change. Now, we could have, there could have been a substitution that instead of a C, it could have been an A, right? So if that would have happened, if it would have been an A, and that would have been a C, that would have been a G, and UAG is actually a stop codon. So you would have had a shorter protein chain compared to the original, which would have made the protein different, which would have made the trait different. So that's just an example of substitution, how in purple it would have kept the protein chain the same, or the amino acid sequence the same, but if I would have made that change that it did in green, then it could have had a completely different effect on that amino acid sequence in that protein, right? Okay, so now let's do the insertion mutation. And remember, insertion is where they add a DNA base into that original sequence. So let's go ahead and do our new DNA sequence with that insertion. So we have ATG, CGA, TTA, CTG. Alright, and so if you notice, we have inserted this extra T in. Okay, so going through the process, we're going to replicate it to make another strand of DNA. So then, therefore, that would be TAC, GCT, AAT, GACT. Right? And then if we were to transcribe it and make that mRNA strand, TAC would be AUG, GCT would be CGA, AAT would be UUA, then GAC would be CUG, and the T would bind to an A. Alright, and then if we did our codons, right, and we would just box them around until we ran out of them. Alright, so we would have AUG is going to be MET, then CGA, so CGA is still arginine, then UUA is going to be UUA is going to be leucine, and then CUG is also going to be leucine. So if you compare the original to the new strand, we still have our MET, we still have our ARG, but instead of a tyrosine and a stop, we have two leucines. So because this protein change, or the amino acid sequence is changed, that changes the protein, which changes the trait. Right? And then so if a trait changes, it could either give you a disease or it could prevent you from having something. So it depends on what the gene is. It depends on what trait it controls. But like, for example, if you have a different protein chain for breast cancer, okay, then from the original one, if you have that altered one, then you would be more susceptible to getting breast cancer, or you would develop breast cancer, versus not having a that affected amino acid chain and sequence. All right, and then the last one that we are going to talk about is deletion. All right, and deletion is where you have one of the bases is removed. Right, so we would have a DNA sequence of ATG, CGA, ACTGA, right, and then that would mean this T was removed, right, and then so we would do the same process where we would replicate it so that ATG would bind to TAC, TAC, CGA would bind to GCT, ACT would bind to TGA, and then you'd have the G and the A would bind to C and a T. Right, and then we would have mRNA, and so we would still have AUG, CGA, we would have ACU, and then we would have GA. And then if we boxed them, 1, 2, 3, 1, 2, 3, 1, 2, 3, right? And then if we made our amino acid sequence, we would still have MET, CGA would be arginine, and then ACU would be threonine, right? And then so because the amino acid sequence is different in the deletion sequence than the original, then it would alter the protein, which would alter the trait. And that is it. Good luck on your quiz.